I'll be introducing Dr. Elena Blanco-Suarez. Dr. Elena Blanco-Suarez is an expert in the study of how astrocytes contribute to the protection of neurons following stroke. Originally from Spain, Elena obtained her doctorate from the University of Bristol with the support of the Marie Curie Fellowship Program. During her PhD, she studied how neurons from different areas of the brain respond to oxygen loss and sugar deprivation. After graduating, Elena moved to California to work at the Salk Institute. Recently, she started her own lab at the Thomas Jefferson University, where she aims to understand how astrocyte secretions might be involved in neuronal vulnerability to stroke. Outside of the lab, Elena is committed to furthering science communication and outreach, with a special focus on reaching Hispanic communities. Following this work, she was awarded the Next Generation Award by the Society for Neuroscience and was named a finalist for the AAAS Early Career Award for Public Engagement with Science. Welcome, Elena.
Okay, well, thank you so much for having me, especially with Jen and Chris, that, they have such beautiful stories. So let me share the screen. And thank you. What? Can you see the screen right now? Yes, right? Okay. That's the question that we've been asking for the last year, right? With this pandemic. So yeah, thank you so much for the very nice introduction. As YC said, I just started my lab like literally 6 months ago, barely 6 months ago. So today I'm going to explain to you what astrocytes do after stroke, but I want to focus on something that I started working as a postdoc and what helped to launch the work that I'm hoping to do as an independent researcher, assistant professor now at Jefferson University. So literally what I'm going to show you is nothing that I've done here yet because until yesterday, actually, I didn't have anyone in the lab. It was just me. So it's a work that I've been doing for the last year. So just to start, just a few facts about the stroke in the United States. Every 40 seconds, someone suffers from a stroke, and 1 in every 19 deaths are actually caused by stroke. This is data from 2017, but it hasn't really changed that much today in 2021. And in fact, 87% of the strokes that are at least reported are ischemic, which is the type of stroke that I study, that I'm most interested in. This kind of stroke, like Jen, I'm not that used to see MRIs of human brains. I'm actually studying the mouse brain as the model for our research. But I wanted to show you how it looks in our brain if we have an ischemic stroke. So this is the plane that we are looking at here. And this white, very bright white area here is what represents the area that's been affected by ischemic stroke. But what's ischemic stroke? This is This is a condition that is characterized by an interruption of the blood flow to a particular region of your brain. Here in this cartoon, we see it here in gray, and that will correspond to this white area. And that's what we call the core of the injury. That's the area that's directly deprived from blood, from the energy that the cells need to carry on their normal metabolism and really live. So it's very interesting because actually in research in stroke, we are more interested in the narrow area that surrounds this core of the injury, which is what we call peri-infarct area. We are interested in that region because actually that's where it holds a lot of potential for recovery. Basically, that peri-infarct area is filled with cells that are not completely deprived of blood flow, but they suffer from certain degree of deprivation. So there are going to be many mechanisms that are going to be triggered there, and it will determine whether the cells survive or the cells die in following days and weeks after having a stroke. And after having a stroke, there's actually a progression that is that it can be divided in both human and in mice in these different phases that are listed here in this diagram. And I'm gonna walk you through it because it's very important to actually for the research that I want to do, the research that I'm doing, to know which phase we are talking about. So the first phase that we encounter is the acute phase, and that's right after you have a stroke. Here, you see that there's a cell death, which is what corresponds to the score of the injury, and that happens especially during the first minutes or hours after having a stroke. This also coincides with the time window where when someone ideally will go to the doctor, get medical care. But unfortunately, you all know that that's not always going to be possible because some people, they live alone, they won't even realize that they're having a stroke because ischemic strokes don't have to be necessarily this big. They can be a lot smaller and still have very bad consequences for the patient. This treatment that that it's available right now for ischemic stroke coincides with that time window, as I said, and it's usually the first 4.5 hours. And the treatments that are available is this thrombolysis or recanalization, which basically aims to remove whatever is blocking the blood flow to get to that brain region. Not many patients actually are successful at get this treatment. And when they get the treatment, not always the outcomes are as good as you would expect. So after those first few minutes and hours, the inflammatory mechanisms come in. There is a phenomenon called scarring or the formation of the glial scar, where astrocytes actually have a very important role. And then this is gonna come down and the subacute phase starts. This is a very interesting phase, and that's actually the one that you would say that we're going to be focusing on in the lab, because it's a phase where there is a peak or an increase in endogenous plasticity. This is the natural plasticity that the brain can display. And for those who are not very familiar with the concept of plasticity, this is basically the ability that our brain has to adapt and to change depending on different experiences. These experiences can be signals from the environment, from your own body, and of course, in response to injuries such as stroke or traumatic brain injury, or in response to disease, to any kind of disease. So this plasticity, the human brain has an extraordinary ability, but sometimes it's compromised by different factors. For instance, age, the presence of another concurrent disease that you may have, etc. And the mice have like a remarkable ability to recover to whatever we do to them. So it's a very good model in order to understand what kind of mechanisms are going on here that are promoting this plasticity. And then how efficient this subacute phase will determine the outcomes during the chronic phase. So what I'm doing in the lab is hoping to understand how astrocytes can regulate plasticity during this phase, how we can use astrocytes after inflammation, how these astrocytes are going to be involved in promoting this plasticity. So as you know by now, after Jen and Chris's talks, in the brain, we find different cell types alongside with neurons. I'm not going to argue that neurons are not important. You probably won't make it too far if you didn't have any neurons, even though some people, sometimes it feels like they don't have many, but everyone has the neurons. The neurons are very important, and so are oligodendrocytes, microglia, and of course astrocytes, which is you could say my favorite cell type. So astrocytes are very close with neurons. They do a lot of things. I liked a lot how Jen depicted the astrocytes as little clouds, like they know everything that's going on, as she said. And they actually have a lot of functions that are very important. So some of these functions are like the physical and metabolic support that they give to neurons, how they maintain the very delicate balance that we have in the brain. And of course, they produce these pro and anti-inflammatory responses, the scar formation in response to injury or disease. And they can also reuptake excessive neurotransmitters that might be released in response to different things, for instance, ischemic stroke. And my favorite function is how they modulate synapses. These are the neuronal connections. So during neuroinflammation, astrocytes undergo a lot of different changes, molecular changes, morphological changes, functional changes. And these are all grouped together under the term astrogliosis. As you can see here in this diagram, astrocytes receive a lot of signals from surrounding cells, but they also send signals to their other cellular partners that are there in the brain. We know that neuroinflammation is a multicellular response and not the responsibility of one cellular type or one single molecule. It is a very complicated cellular response, as you might have guessed after seeing all these talks. And actually, in the context of stroke, this astrogliosis can go too wild, let's say, get very severe, and that's when the glial scar is formed. So this is a very pretty picture that I didn't take, but it's very obvious how here the regular morphology of astrocytes is modified, and they become very compact in order to form that glial scar. So the question then it was whether this glial scar was good or bad. And it actually prevents further inflammation. As you can see here, it separates very clearly these two areas. So it prevents that chronic inflammation signals maybe from going further into healthy tissue that is away from the core of the injury. And so it has the ability to limit that injury. But there were reports, and there's still some level of maybe controversy going on, saying whether this glial scar may also block repair. So are astrocytes good or bad? The truth is that they are both. It's not like they are friends or foes. It's not that simple. And what we're trying to do in the lab is to figure out how we can make them friendlier and less of an enemy, at least in the context of injury, of ischemic stroke. So we were focusing, as I said, in how to boost the plasticity, right? And we want to use what we know about astrocytes, about their ability to modulate synapses, neuronal connections, and therefore modulate plasticity, to promote that during stroke. So we know now, after years of research from the lab where I did my postdoc, but also many other labs for decades, that astrocytes can secrete different proteins that are going to modulate that the formation, the maturation, and the maintenance of these neuronal connections. So for instance, here, we're just showing a couple of these proteins. There are many more that have been identified, the thrombospondins and glypicans that are sent to the neuronal connection, to the synapse, as instructions, let's say, to tell the neurons which kind of receptors they have to express there in order to understand the signals that come from the other neuron. So one of the things that happens in stroke is that you lose those connections. There are a significant decrease in the number of synapses. This data corresponds to a mouse brain, but it's something that's been seen also in humans. And as I said earlier, the mice can recover better than humans to a lot of ugly things that we do to them. And one of the things is recovery from stroke. But still, you can see that they lose a significant percentage of these synapses after stroke. And then you see that they recover, which will correspond, if you see at the days post-injury, that it corresponds to the subacute phase, when the plasticity increases and there is more chance to recovery. So, of course, if you lose those synaptic connections, the neurons eventually are going to die. And I think we all agree that a dead neuron is not a very good neuron. So one protein at a time, we're trying to figure out how to use astrocytes to play those roles of formation and maintenance of synapses in order to aid the brain to almost self-repair during the subacute phase, and then having better outcomes during the chronic phase. So during my postdoc work, so the last few years of my life, I've been dedicated to study this one protein, chordin-like one. And what I found is that chordin-like one is secreted by astrocytes. It's very specific to astrocytes. It's not so much in really any other cell types. And when chordin-like one is secreted from astrocytes, there was a maturation of synapses. And actually, when synapses are mature, there's a limitation of plasticity. And this can maybe sound a little bit counterintuitive, because I just told you that it's great to have enhanced plasticity because then we can recover from anything and it's great. But actually, if you think about it, you want your synapses to mature because that's the way of reaching a healthy adult brain and a functional body, really. So this is a very important process that you want to maintain. However, our goal is, what if we get to eliminate chordin-like one at the times that we need it, so during the subacute phase, and then we can increase plasticity even more to aid the brain to self-repair and to prevent the detrimental effects of ischemic stroke. So that's what we're trying to do. I'm going to show you some data that I have, and I did this in the mice brain. So you can see I gave them small strokes here and then looked into the peri-infarct area. And what I found is that in both acute phase, so this is 1 day after the stroke and 7 days after the stroke, so during the subacute phase, I saw that there was a significant increase in the amount of chordin-like one. So that would be this little white signal, white puncture that represents the amount of protein, the amount of RNA actually that will translate into the protein. So when I saw this, I thought maybe then this is what we have here is one of the bad roles that astrocytes play in response to this, from stroke, because if they are increasing the production of this chordin-like one, it may mean that it's limiting plasticity and therefore recovery. So then what we went and did was to take regular mice, and we gave them strokes, and we saw, as expected, that they lost neuronal connections. We measured that by looking at dendritic spines, which are these little, I hope you can see them, mushroom-shaped structures, which is where synaptic mechanisms are normally located. And we saw, as I said, what we expected, that the mouse lost a significant number, almost a 50% of those spines. However, when we took the mouse, completely normal mouse, but without chordin-like one, so we were hoping that brain, we knew that brain was more plastic, like the plasticity was enhanced already. When we did the stroke to these animals, we saw that actually they didn't lose any spines. And this was also true for when we analyzed them during the subacute phase. So this was right after the stroke, and this was during the phase where plasticity is already expected to be enhanced. So these chordin-like one knockouts, as we call these mice that lack the protein, had allegedly even more plasticity. So the absence of chordin-like one, and this is something that we are working on to figure out what's actually going on, is either protecting against complete spine loss after the stroke, or it's a really quick spine turnover, a really quick regeneration of the spines that were lost, because we're looking at 24 hours. So during those 24 hours, that could have happened since these coordinate 1 knockout mice have that increased plasticity. But either way, we think that eliminating coordinate 1 is actually might be protecting brain function from stroke. So that's a very interesting finding, that it's gonna it opens a lot of avenues that now we're gonna be investigating in the lab. Another thing that was very interesting to see was actually that coordinate 1 was pretty restricted to a certain region of the brain. So here again, the white puncta represents the amount of RNA for coordinate 1. And you see that the signal here is much higher than in other brain regions. So we believe that the role of astrocytes regulating plasticity is going to be probably specific to the brain region. And it might be that when you have a stroke, these astrocytes may all be like expressing the same kind of proteins, and coordinate 1 might be also upregulated, increased in whatever area you're considering, or maybe there are other proteins playing that role. It's also very important to consider which post-stroke stage we want to analyze. And there are other aspects like age and sex that are also going to be very important. So just as a take-home message, the endogenous plasticity here that is displayed by the brain during the subacute phase will be influenced by all these factors. And what we want to do is to take astrocytes and whatever they are doing during this subacute phase, hopefully just be able to manipulate to live with only the good roles, just make them, as I said earlier, friendlier, and use them in order to help the patient to more efficiently go through this subacute phase and then avoid the disability or the other impairments that will appear during the chronic phase. So to finish, I want to thank my former supervisor, Nicola Allen in the Salk Institute, and the people who actually helped me with the very initial steps when I discovered coordinate 1 and all that, the people at the Salk Institute and UCLA who helped me. And if you want to follow us for future more data and whatever we found, you can find us. You can find me on Twitter, or you can send me an email. And of course, I just want to say that, like Jen at the end, just be a decent human being. It's just basic stuff here. Okay, well, thank you. That's that.
Thank you so much. Thank you so much. That was really informative. And we do have some questions right here. So the first question for you, Dr. Blanco-Suarez, is, in the cord-like 1 knockout mice, are there any behavioral or anatomical differences prior to stroke?
So we didn't see any anatomical differences between the wild-type mouse and the mouse that doesn't have the coordinate 1. But we did not analyze any behavioral stuff. Like when I was working with these mice, they seemed normal, but I didn't really analyze it. And that's something that we want to study now, because, of course, after stroke, it's kind of important to see what the motor impairments might be. But we obviously have to analyze if we're going to keep using the coordinate 1 knockouts, especially because they are total knockouts. So the entire, for those who don't know technical stuff, they are lacking coordinate 1 in the entire body. So obviously we're only looking at the brain, but who knows if it's affecting some other stuff that we haven't looked into. So that's a very good question. That's something for the future. Yeah.
Okay, thank you. Sorry, you cut off a little bit towards the end.
Oh, sorry.
Yeah, no, that's okay. And so we do have more questions, but for the time being, we're actually going to go ahead and save them for the panel, if that's okay. Sure. And so, awesome. Thank you so much again, and we'll see you again very soon.
Thanks.
Yeah, no problem.